Next, let's invite Yang Bing for your presentation. And can everybody hear me? And can you see my slides? Okay, thank you. I'd like to thank SSC for this opportunity. I am Yang Bing. I currently study at China Agricultural University. And I may begin. My supervisor is Professor Zhou Xin, and I studied my master's at Capital Normal University. My supervisor was Professor Zhang Aibing. My topic is using artificial intelligence to identify defined species and track the genealogical origin of geographical populations. And this is the first part, using artificial intelligence for species identification and delimitation. The work this is something I published as systematic biology, and I was honored to be selected on the cover of the current issue. And by integrating molecular and morphological data and using deep learning algorithms to achieve more accurate species identification and definition results. Accurate species identification is the basis of biological research. And currently, there are two common methods, traditional morphological methods and DNA barcoding methods. And identification based on morphological characteristics is intuitive and simple, and barcoding is supplemental to morphological methods, which avoids the problems of relying on subjective judgment. And both methods have their own advantages and disadvantages. For example, morphological methods are susceptible to phenotypic plasticity, seasonal dysmorphism, and sexual dimorphism. And the slides on the slide, the upper left shows the two-type transformation of migrating locusts, and the same individual appears. The lower left shows the seasonal variation of junior almona, and with the picture on the right shows the sexual dimorphism of Papulopolitis. And taxonomic knowledge is required in such research, and you can use to look at the features of the wings of the butterfly and the veins are used in recognizing and targets. In recent years, AI has received widespread attention in all weather field monitoring and recognition, and using cameras to capture. And sometimes the identification would make mistakes and there are errors. For example, cats are recognized as dogs, and the confidence level between the two is quite different. And I found on the online that the pictures during the day are pretty different and quite obvious to the human eye. So automatic recognition has their own limitations. And for example, it's affected by the difference in the samples, the specimens, the camera angles, the scale loss, or incomplete wings, etc. And the accuracy is low. DNA barcoding has many advantages and certain limitations as well. For example, the presence of pseudo genes would lead to an overestimation of the number of species. And incomplete lineage sorting will lead to the transformation of polyphyletic groups in polyphyletic group analysis. And Jeffrey Hinton, a Turing Award winner, proposed the concept of deep learning, a branch of machine learning that includes multiple hidden layers and describe the attributes and characteristics of objects in a deeper level, including an input layer, hidden layer, and output layer. And the complexity of the network structure provides the basis for extracting morphological and sequence features simultaneously. In recent years, more and more scholars have proposed integrating genetics, physiology, ecology, morphology, etc., to understand the process and trend of speciation and integrated taxonomy to have more accurate identification and provide more information, including multiple dimensions of samples, including form, sound, molecular evidence signals as well. And we divide the test data set into coarse-grained data set, fine-grained data set, and genomic SNP data. And with the coarse-grained data set includes multiple families and genera, including butterflies, moths, and fishes. And for fine-grained data sets, are closely related species of the same genus. And genomic SNP includes bees and locusts, which are identified as subspecies levels. And for the coarse-grained data, we included 123 species of beetles, 24 species of butterflies, 150 species of moths, and 214 species of fish. And all the samples are from either our labs or Bolt system websites. For fine-grained data set includes 5 closely related species of the same genus that are pretty similar to the eye, and they're similar in morphology and difficult to distinguish, and they share similar gene fragments, including COIC, CYTB, F1 alpha, etc. The genome SNP dataset includes 2 species and 8 subspecies of locust, Corthypus, and 8 subspecies of Apis cerana. To use machine learning to capture features of DNA sequences, we need to encode DNA sequences into matrix by one hot. And there's only one position in each vector contains an information site. And all the other sites are zeros. The training data set increased from 1 individual for each species to 4 individuals to each species, and the test data set is always 1 individual of each species. And this is our MNet morphology molecule network. And up on the upper part is an input of images and sequences on the lower end. And the input data passes through the convolution layer to obtain the extracted features and then sent to the channel module to focus on the meaningful input of data. I think the slide's a little bit stuck. So through the max pooling and I-squared curve, and then there will be an outcome. And all these different layers could come up with more accurate data. So I will focus on all these different layers of blocks. We noticed that in neural networks class taxonomy, the networks would slack off. For example, you'll pay attention to simpler information rather than the bioinformatics that we want the network to focus on. So we need to add a convolutional attention module to help the machine learning. Research shows that a slight movement of the image will significantly affect the confidence of neural network output. And on the left of the bar graph shows that figure represents the output of the baseline network. We thought the image didn't change. There's only a slight move of pixels, so it seems unchanged to the human eye, but the neural network views such the changes, and the accuracy would from the red is a low recognition rate, and on the bar chart on the right shows with the convolutional attention module, and the accuracy will improve. So we have the with the added layer, the accuracy will increase. And the pixels difference is that because the dimensions of the size are not uniform. So with pixel wide is 128 multiplied by 128, and for the sequence there will be different dimensions with 658 multiplied by 15 multiplied by 1. And by incorporating all these different matrices, and the size is uniform to 156 multiplied by 156 plus 1 divided by 2, and there would be a covariance matrix module. And the performance of the MMNet is shown here. On the left, that's the fuzzy ID algorithm, shows a reduced false positive. And on the right, that's BP. And it's the first AI method designed for DNA sequence species identification. And the upper picture shows the result of using an optimized VGGNet, which increased the depth of network and significantly improved accuracy of the network. And the picture on the lower end is of AlexNet. And here are the results of our research. We have looked at sequence length and for example, the length of barcodes, and we chose to be half of the original length because that's the most consistent with most museum samples and collections. And for the image size, we trained all the way from 128 all the way to 512, and we also increased other parameters as well. And this shows the impact of image size and sequence length on MMNet from 128 all the way to 512, and shows that MMNet has higher accuracy under different image sizes. And MMNet is more robust to sequence length. The volatility is lower compared to VGGNet. So MMNet is more robust compared to VGGNet. Increase in training set data will help the model to learn about more diverse features. And here you can see that with the increase of numbers of data, individual data sets, and the accuracy would increase. And even with smaller sets, it can still achieve baseline so that users do not have to collect a vast set of data to achieve quite a high level of accuracy. And these show two different sequence lengths. It shows that the model is also robust in terms of sequence length as well. And we tested learning rates, and we increased by from 1 to 7. And learning rates is similar to stride size. So if the value is too large, local minimum will be missed, and the value is too small, it will take more time to converge. So it's really relevant to performance. And here you can see the axis of accuracy and iteration. And from the value goes down and the accuracy will go up. And originally the learning rates is set, and our model can adjust to our model is adaptive and can adjust the learning rate on its own. The receptive field is similar to visual range. With larger receptive field, the more related objects can be seen. But a larger field would increase the amount of calculation. And from the graphs here, we can see that when the receptive field size is 7, we can have an optimal results. And these graphs show our methodologies using sequences. And so the red bars and the blue ones using BP and fuzzy ID algorithms, and purple and blue columns are AlexNet and VGGNet, and the yellow bar is MMNet. So compared to other algorithms, our accuracy is significantly higher, and other than higher than the algorithm using molecular or morphological data. In terms of fine-grained data sets, the MMNet has a higher accuracy in EF1 alpha and multi-gene fragment data sets. than BP, fuzzy ID, and VGGNet, and we can provide more comprehensive information that are important. TSNE is a dimensionality reduction algorithm. Points of the same color in this represent different individuals of the same species, and our network can cluster different individuals of the same species together while distinguishing individuals of different species well. And in terms of species definition or identification using algorithms, there is no universal threshold, and that can vary. And in MMNet, we calculate F1 values to determine whether the current test sample matches the species in the database. When the database is large enough, it can also help taxonomists quickly locate suspected new species. The average confidence level of the test results of non-beetle data set is 23%, which is well below the current database specific optimal threshold, 76%. And then I'll talk about the background of the research. Origin or traceability is very important. For example, we need to find the origins of traditional medicine and to distinguish between the products that is artificially cultured or poached. And biodiversity investigation is a prerequisite for protecting the genetic diversity of species and to prevent invasive pests. So tracing originality is very important in so many different aspects. And in the work, traditional phylogenetic framework is required. In machine learning, a rapid growth provides unforeseen opportunities. Test data set for our research is from 8 geographical populations of oriental bees in China, and that are closely related to subpopulations of the same province. And then research results in terms of accuracy, we can find the FST values and can be reduced calculation time and increase accuracy by 15%. And for the impact of hyperparameters on accuracy, our model is robust to both batch size and epoch, and it's easily affected by learning rates. So after building the model, we allow the model to adjust learning rate automatically. And our model, while testing, can be 300,000 times faster than traditional evolutionary tree methods at test time, and the accuracy is far better than the latest transformer-based algorithm with accuracy over 98%. And our model's interpretability and credibility, and whether it provides insights, are very important. So, and these are the visualization of the top 50 SNP sites of the model. And you can see that other than Bomi and Taiwan populations, this model is leading. So from this graph, combining multiple loci can better distinguish between different populations. To summarize, we constructed a multimodal deep learning algorithm model, and it has high accuracy in distinguishing closely related species, which can assist in judging new species. And when the species size is large, we can help by learning by multimodal voting, and we can be more objective in taxonomy, and it achieves a full process fusion integration rather than an independent judgment at each stage. Secondly, deep learning algorithms have better results in accuracy and computational efficiency. And we learned that the SNP sites and the information sites is affected, but we can improve the predictability and interpretability and credibility of the model, and to find the monitoring sequences so that we don't have to use SNP data all the time. And now we have a lot of big data, different sources of data, including video, point clouds, and climates and biosensors, and that can all benefit species taxonomy and identification. In the future, our model provides a basic preliminary test for future integration and to help with better species identification and to protect all the lives on Earth better before the species become extinct. Lastly, I'd like to thank uh, my institutes and all the teams providing assistance for me. Lastly, I'd like to thank SSE for this opportunity. Thank you. And welcome any comments and critique, and feel free to contact me via email. Sorry, you are on mute. Thank you to Yang Ping for his presentation. It's very interesting. A lot of audience members are already asking questions. And feel free to ask questions now. We're now open to the floor. Do we have any questions? I see there are several questions on the chat. Okay, sure, I'll start. Okay, so Zhang Kai asks the topology and the molecular data, the weighting distribution. We use plants. So the plants taxonomy is easier compared to animals because they don't move comparatively. And the quality of image caption and identification is better. And also for plants, which phenotypes do you use? Do you select flowers or leaves, or do you have any preference? No, we don't have a lateral comparison yet. We would prioritize flowers because the flowers provide genitalia and leaves are included. We didn't separate flowers and leaves from one another. So this is a very interesting question, and I may dig into this further. Another question is that for your algorithms, topology, do you have a weighting distribution for the morphology or molecular data? Well, we allow the model to learn by itself. Allow me to show you the exact slide. Please give me a moment while I share my screen. As you can see, for the Urbanus esta, for these species, the DNA, their contribution to DNA or image are different. So we allow the network to assign weighting on its own. We feel that this can be more optimal compared to all the different species of the same genus. Then how about other classifications? Would the weighting be different? Yes. As you can see, there are different weighting, slight difference in the weighting compared to the species. Okay, thank you. There are also other questions. I'm sorry to interrupt, but because we are a little bit delayed, so I will type in my answers. Thank you. Thank you, Mr. Xu.